the central dogma of the molecular biology. What is the first step into the central dogma of the molecular biology? From the double-stranded DNA molecule, there is a synthesis of the single-stranded mRNA molecule with the help of RNA polymerase enzyme. And this process, it is known as the transcription. Then second process of the central dogma of molecular biology, it is from the single-stranded mRNA molecule, there is a synthesis of the proteins. Proteins are synthesized with the help of the ribosome and tRNA molecule. Okay? This step or this process, it is known as the translation. So one by one, we are going to discuss transcription and the translation, how it happens into the prokaryotic organism. First, let's see about the prokaryotes. You have before that, as we discussed about the structure of the double-stranded DNA molecule, you know the both the DNA molecule have the two strands. One strand runs in the 5' to 3' direction, while other strand runs into the 3' to 5' direction. Okay, and to this both the strands at the central axis, the base pairs are attached perpendicular to the central axis. Okay, and this in this base pair, adenine pairs with the thymine, and guanine pairs with the cytosine. Adenine pairs with the thymine by forming two hydrogen bonds, and guanine pairs with the cytosine by forming the three hydrogen bonds. The DNA molecule has two grooves, minor groove and major groove. Okay? If it is in the structural, these are the structural properties of this DNA molecule. One turn of the double helical DNA molecule, there are presence of the 10 base pair. Distance of the one turn of the DNA molecule, that is the 36 angstrom. Then the distance in between the base pairs, that is 0.34 or 0.36 angstrom. Like this, this all we already discussed into the structure of the DNA molecule, that is proposed by Watson and Crick. What RNA molecule is the single strand? What are the differences into the DNA and RNA that we are going to see? DNA is the double stranded, while RNA is the single. Okay. The bases are present into the DNA and RNA. There are presence of the adenine, guanine, and cytosine in both the molecule, but in place of the thymine, uracil is present into the RNA molecule. Uracil presents here in place of the thymine. Thymine is absent into the RNA molecule. So another important difference in the sugar molecule, here in deoxyribose sugar presents into the DNA molecule, while ribose sugar is present into the RNA molecule. So these are the three important differences, three major differences. Remember, first difference is the DNA is double stranded, RNA is single stranded. Second difference, DNA contains the deoxyribose sugar, RNA is composed of the ribose sugar. In DNA, presence of the thymine base, and in RNA, presence of the uracil base in place of. So these are the three major differences in between the DNA and RNA molecule. So after that, we are going to see about the transcription. What is the transcription? From the double-stranded DNA molecule, there is a synthesis of single-stranded RNA molecule, and this process, it is known as the transcription. This process, it is carried out by the enzyme RNA polymerase. polymerization of the RNA takes place onto the one strand of the DNA molecule that we are going to see in next slide. So ribonucleotides are added. Okay, opposite ribonucleotide or complement ribonucleotides are added during the synthesis of the RNA molecule, and this RNA molecule. So then RNA molecules are of the three types. These are with the help of nuclease enzyme, that is RNase enzyme, makes the cuts into the RNA molecule. Some RNA are mRNA, some are the rRNA, that is a ribosomal RNA, which plays role in formation of the ribosomes, and some RNA are the tRNA molecule, which plays important role into the synthesis of protein. Then into the double-stranded DNA molecule. This double-stranded DNA molecule, how the synthesis of the RNA or how the transcription this process takes. Transcription this process into the DNA molecule. There are three specific reasons are present into the prokaryotic DNA. So the first reason into the DNA, double-stranded DNA molecule, it is known as the promoter reason. So the promoter gene, it is very, very important. This promoter gene requires for or serves as the binding site for the RNA polymerase enzyme that we are going to discuss. From here, the +1 base starts. So +1 base starts of the mRNA molecule. It is the RNA coding sequences. These are all are the RNA coding sequences from this one, +1 to up to this. Okay, and at the last, there is presence of the termination sequences. So here, the termination of the transcription process takes place. So at we can see where the initiation of the transcription takes place, that reason it is known as the promoter or promoter gene, we can say. Then RNA coding sequences are present into the DNA molecule, and then in last reason termination sequence are present. So in this case, the reason 5' of the promoter sequence is called as the upstream sequence. The reason 3' of the terminator sequence is called as the downstream. Before this reason, okay, so it is this process is also takes place. Transcription process also takes place from 5' to 3'. As yesterday we discussed about the DNA replication, it also takes place from the direction of the 5' to 3'. Then reason of the 3' position of terminator sequences are called as the downstream sequences. So here it is of, we can say here the 5', this reason, it is a 5' reason, and these are the 3' reason. Okay, 3' reason, and these sequences are called as up. So that the 5' of the promoter sequence is called as the upstream. So this reason it is called as the upstream, and this reason it is called as the downstream. Okay, downstream reason this way it is called with a specific nomenclature. In next slide, the genes are composed of the 3 regions as we discussed: promoter region, then RNA coding sequence region, and third is the termination region. Then the promoter region, what is the role of this promoter region? Upstream of the sequences that codes for the RNA, site of the interaction of RNA polymerase before making the RNA. So because the promoter region is to promoter region, RNA polymerase enzyme binds to the promoter. Interaction in between RNA polymerase enzyme and promoter region takes place, or the promoter gene is serves as the binding site for the RNA polymerase enzyme. Then the coding region, DNA sequences that will become copied into the RNA molecule, that is known as the RNA transcript, or the production of the transcription process. It is known as the transcript, means the RNA. Then last region, the region tells the RNA polymerase stops to make the RNA molecule. Transcription process involves the three steps. The first is the initiation, second is the elongation, and third is the termination. Where does the transcription initiation takes place? At any genomic regions or locations? No. It takes place only the upstream site of the +1 base, and this site it is known as the promoter gene. So this is the double-stranded DNA molecule shown in the light green and dark green color, and this is the RNA polymerase enzyme. RNA polymerase enzyme, it is various subunits are present into the RNA polymerase enzymes. These subunits are two alpha subunits are present, beta subunit is present, beta dash subunit is present, and sigma subunit is present. Means that total 5 subunits, the RNA polymerase enzyme, it is composed of the total 5 subunits. And this RNA polymerase enzyme binds to the promoter gene. And into the promoter gene, there are the two kinds of the specific sequences are present. -10 sequences. Okay, means from the at here there is the +1 base. Okay, +1 base onto the DNA molecule. So upstream of this site, -1, -2, -3. In this way, the bases are counted. And here at near -10 region, the my these specific sequences are present here into the -10. This is a -10 region, and at this -10 region, these sequences are known as the 5' to 3' TATAAT. This is also known as the Tata box. -10 sequences are also known as the Tata box. And above this upstream side, the -35 sequences are present. These are known as the -35 sequences, and these -35 sequences are TTA, TACA. Okay, so these -10 and -35 sequences plays important role in binding of the RNA polymerase enzyme, and sigma factor recognizes this RNA polymerase recognizes this RNA polymerase enzyme. It is correctly bounded to the region, okay, because it identifies by binding the sequences to the -10 and -35 region. So this sigma factor plays very important role here into the binding of the RNA polymerase enzyme. So this sigma factor binds to the promoter of -30, -10 and -35 region, and core enzyme binds to the sigma factor and promoter, but DNA still closed. So it is called as the closed complex. Okay, closed promoter complex. This hollow enzyme untwists the double strand of the DNA molecule. Then this untwist promoter, it is called as open promoter complex, and then RNA polymerase binds to the -10 sequences and place the position of the start transcribing. Sigma factor is released so that core enzyme can go forward transcribing this or synthesis of the RNA molecule. Okay, so this core enzyme and hollow enzyme, there are the two terminologies here. Core enzyme, okay. So the RNA polymerase enzyme, then sigma factor, it is not bounded to this enzyme, then it is known as the core enzyme. And when sigma factor it is bounded, then it is known as the hollow enzyme. So these two terminologies here. So when the this RNA polymerase enzyme bounds to the promoter region, so these are the -10 and -35 regions, and then after binding of the sigma factor, this closed complex becomes the open. You can see here, two strands of the DNA are open. But here into the RNA, the shear synthesis of the RNA, that is transcription process, RNA polymerase itself does the work of the helicase enzyme. RNA polymerase enzyme itself open the double-stranded structure into the single-stranded form. Okay, so this is called as the open promoter complex. This promoter gene is open, and then at the +1 base. Okay, here this +1 base is present here, and this after binding of the sigma factor, it becomes open complex. And after open complex is formed, then this is known as the transcription bubble, like this. This is the open complex. Okay, and in this transcription, then initiation of the transcription starts. And when the initiation of the transcription starts, it when the RNA polymerase enzyme moves 5 to 6 or 6 to 8 base pair uh, forward, when it forwards the 5 to 8 base pairs, then the sigma release of the sigma factor takes place. And again, from the hollow enzyme, there is conversion of the core enzyme. Okay, so this enzyme without sigma factor, it is known as core enzyme. And then after release, so the sigma factor is only important during the initiation of the transcription. So the function of the sigma factor, which recognizes whether this RNA polymerase enzyme, that is the core enzyme, it is bounded to right initiator region or right promoter gene or not. And that is recognized by the sigma factor of the RNA polymerase enzyme. And then initiation of the transcription process for the 5 to 6 or 6 to 8 base pair. It moves, RNA polymerase moves towards the um, downstream region, as we discussed, towards the 1+1 and +5 and 6. The release of the sigma factor takes, and then one strand of the DNA molecule acts as the template for synthesis of the RNA molecule. Okay, and here RNA molecule, it is a complementary to the synthesizing strand, so that it is while this template strand, while other strand of the DNA molecule, it is known as the coding strand. You might have been observing this figure, and here this is a label. This another strand, it is known as a coding strand, and that strand which serves as the serves strand for the synthesis of the RNA molecule, that is known as the template strand. Whatever the nucleotides are present here onto the DNA molecule, opposite nucleotides are added by the RNA polymerase enzyme. Beta and beta dash subunits of the RNA polymerase enzyme, they do this work, synthesis of the or catalysis of the RNA molecule, and they adds the nitrogenous bases or nucleotide triphosphate, that is a ribonucleotide triphosphates are added here. And if the adenine is present onto the DNA strand, then uracil it is added. If the thymine is present onto the DNA strand, adenine it is added. If the guanine is present onto the DNA molecule, then opposite or complementary base cytosine it is added. And in this way, polymerization of the RNA molecule takes place. You can see here there is a synthesis of the RNA molecule. And synthesis of the RNA molecule always takes place from the direction of the 5 prime to 3 prime. And this synthesis 5 prime to 3 prime, you can see here 5 prime to 3 prime. Hence, the direction of the template strand always 3 prime to 5 prime. Direction of the template strand of the DNA always the opposite direction, complementary strand, that is the 3 prime to the 5 prime. So always remember this information is very, very important for you. As it is open complex, then the nucleotides added. Now, this is the one strand of the DNA as is last slide we discussed. This is the brown color strand. It is the DNA strand, and the how it is synthesized. Yellow strand. It is the RNA strand. It is synthesized into the direction of 5 prime to 3 prime. Okay, 5 prime to 3 prime, and DNA template strand would be the having the direction of opposite direction 3 prime to 5 prime. Okay, and this nucleotides are added like this. You can see here guanine is there, then cytosine is added. In yellow color, that is the RNA molecule. This yellow color, it is RNA, polymerization of RNA. So cytosine is there, then guanine is added. Adenine is there, then uracil is added. So opposite nucleotide or opposite ribonucleotide is added during the synthesis of the RNA molecule. And this polymerization, it is carried out by the RNA polymerase enzyme. So this is the initiation and elongation of the transcription process. That is the first process into the enzyme. RNA polymerase opens the DNA strand and synthesizes the RNA complementary to the one strand of the DNA molecule. And that strand, it is known as template strand. So in this way, you can see the complementary RNA, it is synthesized, and at the termination, the synthesis of the RNA molecule stops here, and both the strands again the reunite. This work is done by RNA polymerase itself. The opening of the two strands of the DNA, then further rejoining of the two strands of the DNA. This work is done by the RNA polymerase enzyme itself. Okay. So the now then how the termination of the this transcription process takes place. Termination of the transcription process takes place two ways. One is the row dependent termination, and another is the row. One is the row independent termination, and another is the row dependent termination. So it is known as row independent termination, type 1 terminator, and this is the row dependent termination, means it is a type 2 process. Okay, so you can see here how this termination takes place. The sequences with the DNA core consist of the inverted repeats to the upstream of the termination point. Suppose here the termination will be there, then the to this upstream process inverted repeats are present here. G, G, P, C, G, like this. Okay, and here G, G, T, C, G. These are the inverted repeats. These are called as the inverted repeats. Or C, C, A, G, C, T, C, C, A, G, C, T, like this. These we can find these are the inverted repeats. And when the RNA molecule it is synthesized after the transcription process, in RNA, these sequences are also, these sequences will also come. So that the G, it is a complementary to the, here the C. This is another G, it is complementary to C. So these are bases are the complementary to each other. And because of the complementary bases, so here what will happen? This there is a formation of double-stranded structure. This is the single-stranded because complementary bases are not there. This reason is the single-stranded. The complementary bases are not. But reason this, we can say 1, and this reason, this is a 2. So this one reason, it is complementary to 2 reason, and because of that, formation of the double strand of the RNA molecule takes. And this kind of the structure, it is known as the stem loop, or it is also called as the hairpin loop. This is the stem loop. It is like the hairpin. Hence, this is called as the hairpin loop formation takes place. So, after the formation of the hairpin loop, the RNA or termination of the synthesis of the RNA stops there. So, if we see the next slide like this, as it forms the loop, the RNA polymerase enzyme, it is bounded, and it release of the RNA polymerase from the double-stranded DNA takes place. So here termination, in this way the termination takes place. Now second kind of the termination we are going to see about the row dependent termination. It is the row dependent termination. Like this row protein is important. The row protein plays important role in the second type of the termination, and this row protein, it is the hexameric protein, binds to the RNA molecule and travels along with the RNA molecule towards the termination sequences like this, as shown in this figure. So this row protein goes and travels towards the RNA polymerase enzyme, and as it touches to the RNA polymerase enzyme, then the synthesis of the RNA stops here, or transcription process stops here in this particular point, and termination of the transcription. So this is known as the row dependent termination. Row protein, which is known as the helicases enzyme also. It is helicases like enzyme. It moves to the location of the RNA polymerase enzyme and row unwinds the DNA and RNA. So whatever here RNA bounded here, it removes the RNA from this. It removes the RNA polymerase enzyme also, and release of the this RNA molecule takes place into the cytoplasm. In this way, the row dependent termination takes place. Okay, so row dependent termination process requires the ATP, because row protein, it is the ATP hydrolyzing protein. It hydrolyzes the ATP into ADP and takes the energy and to stops the termination process. So this is the termination of the transcription. Two way it takes place. First way is the row independent termination, because there is a presence of the inverted repeats, and because of the inverted repeats, as the double strand structure formation takes place into RNA molecule, then this transcription process stops. Then row dependent termination, it travels along with the RNA molecule and goes into the termination site of the DNA, and then it's releases the RNA molecule, releases the RNA polymerase enzyme here, and separates the RNA molecule.